Greetings, class. Today we're going to talk about Chapter 3, the molecules of cells. So I really like this chapter. It's a basic breakdown of the kinds of nutrients and the things that we take into our bodies pretty much on a daily basis to make sure that we are well nourished. So the main big ideas that we're talking about for this chapter, we're going to have an introduction to organic compounds. And organic compounds are typically things that contain carbon. We're going to talk about carbohydrates. So whenever we think about carbohydrates, think about, let's see if I can get my little pen to work. So when we think about carbohydrates, think about sugar. Let's see if I can draw on here. Here we go. So think about sugars. So things that taste good, maybe some of your favorite foods, pastas, breads, candy, anything that tastes sweet. And for example, right here we have honey. These carbohydrates are providing energy for our body. Next up, we have lipids, and lipids are also known as fats. Fats are good to have for your body. Of course, we don't want to have too much, but it's always a good idea to have fat because it helps us to make certain hormones. It helps to keep us warm. All right, next we have proteins. Proteins do a ton of things in our bodies, and we'll get more into it in a little bit, but proteins do a lot of things. So it's good for us to have protein as well. Some examples of protein that we might have in our diet, of course, meat, so meat from animals. And then also for those of you that are vegetarian or vegan, proteins that you can get from maybe tofu or beans. Lastly, we have nucleic acids. So when you think of nucleic acids, think of DNA. DNA is our hereditary material, and it is passed from parents to offspring, to make sure that we carry on the lineage. All right, so let's talk about our organic compounds. So life is so diverse because it's based on the properties of something called carbon. Carbon can form 4 other bonds with other atoms, and it is the base for filling pretty much a large, diverse array of organic compounds. So carbon chains, these form the backbone of most organic molecules. So for example, isomers, they have the same molecular formula, but different structures. And lastly, hydrocarbons are composed of only carbon and hydrogen, just like they sound, right? So hydro and carbon. So the main thing to pay attention here is that most of life's diversity is based on carbon. Most living things are going to have a carbon base, right? And we're going to have isomers and hydrocarbons. Go ahead and look at those definitions, write those down. Okay, so for example, we have ethane and propane. These are examples of hydrocarbons. Okay, just some more pictures. All right, so a few chemical groups are key to the functioning of biological molecules. So an organic compound's properties depend on 1, the size and shape of that carbon backbone. So that's number 1. And then the different atoms that are actually attached to that kind of backbone. So things that are hydrophilic, so hydro, referring to water. So let me put hydro right here. Water, H2O, right? And then philic, so this word right here, P-H-I, I'll draw that in. Philic, this means loving. Okay, so anything that's named hydrophilic, it likes to be around water. It loves to be in water. So if something is hydrophilic or has hydrophilic something called functional groups, these are going to give those organic molecules specific chemical properties. And we'll look at some more of those. Okay, so for example, we have different things called sex hormones. So testosterone and estradiol, which is a form of estrogen. Typically, genetic males are making more testosterone and genetic females are making more estradiol or estrogen. So these are the main types of sex hormones. So when you think of testosterone, you might think of that injectable that you get, but all humans typically make testosterone, just genetic males are going to make a lot more. Okay, so for example, there's only a few differences. We can see right here, right? We have something called hydroxide on this last little circle of estradiol. And then for testosterone, we're just having a lonely little oxygen by itself. Okay, and then another functional group right here. So you see pretty much the same chain. It looks the same for this sex hormone chain, but just a few key differences are going to make the differences between those sex hormones. All right, so cells make large molecules from a limited set of small molecules. So there are 4 classes of biological molecules that I mentioned towards the beginning of this lecture. These are all made from single units, something called a monomer. Okay, so just think of 1 unit or 1 piece of something, right? So let me draw, it says 1 unit, and I'll put just 1 little circle. Okay, so that is a monomer. So mono means 1, right? So 1 piece of something. So everything that's larger is made up of many, many monomers or many, many units. So the larger molecules that are made from these monomers can be called macromolecules because they're large. So macro meaning large, and then referring to the molecules. You can also call these larger particles polymers. So poly, so P-O-L-Y, also means many. So because they are made from identical or similar building blocks kind of strung together. So here's our monomer, right? And then you put a bunch of monomers bound together. We can make a polymer, right? Which is also known as a macromolecule. So monomers are typically kind of linked together to form polymers, right, through something called dehydration. So think of a dehydration. What's happening? You're probably really thirsty, lacking water, right? So in order for these monomers to be linked together, they have to lose water. So let me go ahead and draw my little circle again. And this little circle is trying to link to the next circle. But what has to happen first is that you have to take away some water. And that's that dehydration reaction. It's taking out water. So now these 2 little monomers can form a link together, and it keeps on going down the line to get that polymer. Okay, so because we take water out to form a bond in order to break a bond between these polymers to kind of take them back to monomers, we have to do something called a hydrolysis reaction. And this is when we're using water. Okay, so I'll put H2O. Remember, hydro means water. And anytime you see this word lysis, L-Y-S-I-S, that means to break apart or destroy something. So for example, for this part, we're going to take in a water molecule to destroy or break this bond. So now what happens, we added water in, we break the bond, so that polymer is actually broken down to its singular parts. A lot of times for these types of reactions, we have something called enzymes that help it to happen. All right, so for example, we have a short polymer and just a single monomer. It's not really linked to anything. Okay, so then we have a dehydration reaction. Remember, dehydration, we're going to lose this water molecule. And now the bond between this short polymer and this single monomer can now come together. And now you have this longer polymer. Remember, poly means many. Okay, so kind of going the other way. So now we want to break those bonds between each other. Now we're going to do a hydrolysis reaction. Remember, water and then breaking. So hydrolysis, using water to destroy or break something. So you see right here, we're adding in this water molecule. It's going to go in here and break apart the bonds. You see right here, it's going to break this one. And now you have this short polymer and the single monomer again. Okay, so think of water when you're kind of thinking of joining things together or breaking things apart. All right, let's get into the 4 macromolecules of life. And we have 4 of those. Remember, macro means large. So typically we're going to talk about these different classes. They're macromolecules. So the carbohydrates, the lipids, the proteins, and the DNA, those are known as macromolecules. Okay, remember, first off, we have carbohydrates, and I love these carbohydrates. They taste so good. This is sugar. So things like pasta and bread and candy, all the things that taste great, right? So carbohydrates are also made from monomers. So like we just talked about, monomers and polymers, those are going to refer to these different types of organic compounds that we're going to talk about today. So carbohydrates, the most simple form of a carbohydrate is something called a monosaccharide. And we can use the term monosaccharide and this sugar monomer or just monomer kind of interchangeably. They equal the same thing. So remember mono, so M-O-N-O means one. Okay, and then saccharide we have right here means sugar. Okay, so that means you have one sugar. So typically carbohydrates are usually going to kind of range from that small single sugar molecule, which is a monomer, to larger ones called polysaccharides. Remember, poly means many. Okay, and I'll just put many. Oops, that looks weird, but we'll say many sugars, right? Because saccharide means sugar, and I'll put sugar up here. Sugar. So a monosaccharide is one sugar, while a polysaccharide is composed of many sugars. Okay, here's our little bees in the honeycomb making honey, which is a natural sugar. All right, so let's say, for example, let's take two monosaccharides, which are the exact same thing as monomers, right? So let's say one monosaccharide and another monosaccharide to make those two monosaccharides. We bind them together, right, by taking away a water molecule in our dehydration. Remember, you're kind of getting thirsty, right? You don't have a lot of water. We're dehydrating this to make a bond between these two monosaccharides. And now that they're bonded together, so we have two, it's now known as a disaccharide, and that's the term we're looking for right here. Okay, so di means two, okay? So we combine that di with saccharide, and that means two sugars, okay? So di is two, and saccharide is sugars. Okay. And there's another example of glucose. So one glucose molecule plus another glucose molecule is going to equal maltose. And see right here, we have that dehydration reaction because we're taking that water molecule out. Okay, and here's a little graph of the recommended amount of sugar. So for a year, the WHO, which is the World Health Organization, only wants the average person to eat about 20 pounds of sugar per year. The FDA recommends about 40 pounds per year, while the average American is eating 130 pounds of sugar a year. Okay. So when we have all this excess kind of added sugars, they have a lot of adverse health effects. It causes something called systemic inflammation, which can lead to heart disease, cancer, diabetes, all that kind of stuff. Okay, so polysaccharides, we talked about monosaccharides, we talked about disaccharides, and now let's talk about polysaccharides. Remember, poly means many. Poly, that's going to mean many. And then saccharides refers to a sugar. So these are long chains of sugar. So for example, things like starch, if you've heard of starch before, or glycogen, these are known as storage polysaccharides. So just really long chains of sugar. For example, animals like we have glycogen stored in our liver and in our skeletal muscle. And what happens is that in between the states when we're eating, those are called fed states. So think of, I'm feeding my face, right? So fed states. So in between those states, when we're not really taking in any kind of nutrients or glucose or sugar, right? We have to have some way to prevent our bodies from going into something called hypoglycemia, which is low blood sugar. So between those states, our liver is going to actually break down these glycogens, because glycogens are just really long chains of something called glucose, which is a monosaccharide. So it's stored in the liver. And so in between meals, we're going to start breaking these bonds between these single sugars, and they're released into the bloodstream. And that helps to prevent hypoglycemia, or that low blood sugar. Okay. Cellulose is a structural type of polysaccharide that's going to be found in plants. Okay, and then we also have something called chitin, and this is a component of an exoskeleton, so the outside of things like insects and fungi that have their skeletons on the outside. So these polysaccharides can be used for storage, they can be used to maintain the structure of a plant, or they can also compose parts of a skeleton. All right, so for example, you have starch here in the potatoes, right? If you ever heard of starchy vegetables. And these are just really long chains of glucose monomers. So glucose is the type of sugar, it's kind of like the basic sugar, and our body loves glucose because it helps to make energy for our bodies. So remember, monomer is just one, and these are just many, many glucose molecules attached together. All right, so glycogen, we have this, like I said, in our skeletal muscle, also in our liver, same thing, just many, many glucose molecules. You see these? And I'll put glucose down here. Just many, many glucose molecules linked together so that we have a lot of energy stored up and sugar stored up for when we have those states where we're not eating. And the reason why it's stored in muscle as well, so when you're exercising, you have a ready amount of energy to be broken down when you're exercising or just moving around, right? All right, lastly, we have cellulose. Remember, cellulose helps to maintain a cell wall structure. All right, so that was it for sugars. Let's talk about lipids. Whenever we see lipids, think of fats. Okay, so fats, so things that we might eat every day. So things like oil and butter, a lot of kinds of meat that have saturated fat. Okay, so those are your fats. So lipids, we talked about things that are hydrophilic, right? So water loving. Lipids are the opposite. These are hydrophobic. Remember, hydro means water. And if you have a phobia of something, if you've ever heard that word before, and I'll put phobic, that means you have a fear of something, right? So lipids are water fearing. So think of maybe an experiment you did when you were little. You have a glass of water, and then you put some oil on top, kind of shake it up. It looks like the oil kind of mixes for a second, then they separate again. And that because lipids are hydrophobic, they do not dissolve in water. They don't like to be around it at all. Okay, so there are a few different types of lipids. So let's talk about fats. The technical term for fat that we're typically talking about is something called a triglyceride. So this is going to be one glycerol linked to 3 fatty acids. So that makes sense, right? Because tri means 3. Okay, so 3 fatty acid tails attached to one glycerol. So triglycerol. All right, so the fatty acids that have one or more double bonds, those are going to be unsaturated fatty acids. So these are typically considered the more healthy fats, right? They are typical of plant oils. So think of olive oil, grapeseed oil, avocado oil. Those are going to be your unsaturated, more healthy fats. Fats with a ton of hydrogens and no double bonds are saturated fats. These are usually considered the more unhealthy fats. So these are typically going to be your animal fats. Okay. So there is another type of fat you may have heard about, something called trans fats. And these are actually something that the FDA has tried very hard to get out of our food sources, especially fast food, because they can be cancer causing. Okay, so right here we have a picture of a triglyceride. So right here, this is our glycerol head. I'll write that in. So glycerol. You can see that. And then we have 1 three fatty acid tails. And I'll put Fa for fatty acid and then tails. Okay, so triglyceride. Okay, so saturated fats, these are usually going to be solid at room temperature. So for example, butter, saturated fats, you find that in a lot of red meat as well. So those are considered the more unhealthy options. And then to the right side of the screen, we have our unsaturated fats. So our plant oil, this looks like olive oil, right? And then things like salmon or that kind of fatty fish. Those are considered much better for you and much more heart healthy. Okay, so let's talk about a few other kinds of fats that we might see. We have something called phospholipids. And then just like these sound, there's going to be a phosphate and lipids. So it looks pretty similar to a triglyceride, only with the phospholipids, they're only going to have 2 fatty acid tails instead of 3. And of course, they have that phosphate head. All right, next we have steroids. So these include things like cholesterol and some sex hormones. All right, so cholesterol, we have to have this in order to make certain types of hormones, right? So it's a common component in animal cell membranes, and it's also the precursor, which kind of means the starting point for making other steroids, including those sex hormones. Okay. So here's a picture of a phospholipid. So phospho, and then I'll put lipid down here. So just like it sounds, it has a phosphate head. Okay, so here's our phosphate head. And we're going to have 2 little fatty acid chains right here. Remember, these are hydrophobic, so they don't like to be around water, while the phosphate likes to be around water. Okay, so for example, in our cell membranes, typically we're going to have these hydrophilic or water-loving, and I'll put a little heart right here, water-loving heads, and those are facing out towards the environment, that's more liquid, and then facing more inwards toward the cell as well, that's also liquid because they love that fluid. But then you see here we have all these little tails lined up. These are those hydrophobic fatty acid tails. Okay, and I'll put Fa. All right, so I know a lot of you have probably heard about steroids or anabolic steroids. So these are usually going to be synthetic forms of testosterone. And these can be used by different athletes or people who are trying to gain muscle mass and get stronger and more fit more quickly. But they do have a lot of serious health effects. All right, so next let's go on to proteins. So proteins have a very wide range of functions and structures. So proteins are involved in nearly every function in our body, and they are very diverse. So proteins can be enzymes. So enzymes are something known as catalysts. They help to speed up reactions in our bodies. Oops, this will be A, catalysts. Okay, they can function in transport. They can also function in immune defense, so things like antibodies. They can be signal proteins. They can be receptor proteins. They can be contractile, which means they can get shorter, right? So within our muscle cells. They can be more structural, so things like collagen that we have in our skin. And they can also function in storage. So you can see here that proteins do so many things. So proteins are composed of differing arrangements of just 20 amino acid monomers. So thinking about this, a single amino acid is 1 monomer, right? And so we're attaching a bunch of amino acids together, and that's going to form a protein. So proteins are composed of amino acids. So we put my little squares down here. Say this is 1 amino acid monomer. Remember, monomer just means 1. Okay, so we have our little amino acids linked together. And once we have a ton of those linked together, we get a protein. Okay, so proteins' functions are dependent on by their individual shapes, so their 3D structure. In something called denaturation, this is typically due to high heat, acidity, things like that, or different pHs they don't like. A protein is going to unravel and lose its shape, which means it in turn loses its function. So let's talk a little bit more about that. Right here we have a globular protein, just a big glob of amino acids, right? So let's look at how proteins are the same and yet different. So proteins are made from amino acids. So these single amino acids linked by something called a peptide bond. So anytime you see this word peptide, it's usually referring to a protein. So here's one amino acid. Here's our peptide bond. Another amino acid. Another peptide bond. Right? And kind of going so on and so forth. But protein diversity is based on different sequences of those amino acids. So typically they all have a bunch of things in common. So for example, they're going to have an amino group. They're going to have a carboxyl group. They're going to have a hydrogen atom and something called an R group. So an R group is pretty much the only thing that changes between amino acids, and the R group is what kind of gives these different amino acids their differing properties. So amino acids are linked together by dehydration reactions, just like what we talked about with those different carbohydrates, and same thing. So dehydration, we are losing a water molecule. So what happens is that we're joining the carboxyl group of one amino acid to the amino group of the next amino acid, and therefore creating a peptide bond. Okay, so one right here, and we have little side chains. I'll show you a better picture in a second, but just look at these different amino acids bound together. We get that polypeptide, and for each bond that's formed, each peptide bond that's formed, we're going to lose a water molecule. Okay, so for examples, let's look at a couple different examples of amino acids. This amino acid is hydrophobic. Okay, so you see we have a carbon in the middle. We have a hydrogen ion. We have an amine group. And we have a carboxyl group. That's what this means. So these different areas that are kind of highlighted in this orangish color, these are the R groups, and that is responsible for the different types of functionality for these different amino acids. So because leucine, that's this one right here, it has this type of R group, that makes it hydrophobic. The ones that are hydrophilic, it all depends on that R group. And you see they all have the basic things. So see we have our carbon, we have our hydrogen, we have our amine group, and then we have our carboxylic acid. Okay, so all kind of doing the same thing. The only thing that gives them their different properties would be these R groups. All right, so let's talk about the 4 levels of structure for proteins. So a protein's primary structure is just that straight chain of amino acids linked together. So kind of just these little circles, remember that? Just linked together. This is our primary structure. So it's composed of just amino acids and peptide bonds. Next, we have the secondary structure, and that can be found in one of 2 ways. We can have something called an alpha helix, and this is kind of coiling. And then we can have a folding one that's called a beta sheet, and I will show you that in a second. Thirdly, we have the tertiary structure. This is the 3D structure that gives the protein its function, all right? And then quaternary are typically 1 or more polypeptides kind of globbed together. So we have 1 polypeptide, 2 polypeptides, 3 polypeptides, and 4 polypeptides. All right, so let's look at this picture of the primary structure. So each of these little marbles, these represent 1 amino acid, all joined together by peptide bonds, okay? So if you get a question, what type of bonds are found in the primary structure of amino acids, or proteins, they are going to contain peptide bonds. All right, nextly, let's do the 2 types of that secondary structure. So I put a 2, 1 for primary, 2 for secondary. We're going to have something called an alpha helix. And we also have something called a beta pleated sheet. So for the secondary structure, it's found either in the alpha helix or the beta pleated sheet. So these structures are maintained by hydrogen bonds. So if you get a question asking you what type of bonds are found in the secondary structure, you're going to have peptide bonds, of course, and then you're also going to have hydrogen bonds. Next, we have the tertiary structure, and I'll put a 3 right here. For 3 for tertiary, this structure is stabilized by interactions between the R groups, so mainly whether they're hydrophobic or hydrophilic, okay? And so remember, this is the structure, this tertiary structure is what gives the protein its function. So if that protein is kind of unraveled from its tertiary state and goes back to primary, it loses its function in that process called denaturation. All right, so lastly, that quaternary structure, typically polypeptides kind of glob together. So you have one right here and one right here. So 1 and 2. So that's quaternary, just pretty much more than one polypeptide kind of joined together by different interactions. All right, so last type of macromolecule we'll talk about today are something called nucleic acids. So something you probably all heard about is DNA. And DNA contains our hereditary material. Hereditary. Okay, so our hereditary material passed down from parents to offspring. So we have different nucleic acids. Besides just DNA, we also have something called RNA. Okay, so DNA and RNA, those are our main types of nucleic acids. The monomers that make up nucleic acids are something known as nucleotides. So think of nucleic acids and nucleotides. So nucleotides are composed of a sugar, so a pentose sugar, something like this. So 1, 2, 3, 4, 5, because pent, that beginning part, so pentose, it means 5. So we have a 5 carbon sugar, a phosphate group, and I'll put a little P right here. Okay. And something called a nitrogenous base. So it's going to be either A, T, G, or C, and those are going to be the nitrogenous bases, okay? So a few differences between DNA and RNA. DNA is something called a double helix, and I'm sure you've all seen a picture of a piece of DNA before, but here we go. So this is DNA. So you see it has something called a double helix, so one little helix going this way, and then another little helix going up from the bottom this way, okay? So that's our double helix. RNA is just a single polynucleotide chain, which just means it's one straight chain, it's not two. So DNA and RNA are the blueprints for proteins, which kind of are the language for something called our genes. And we always think of something like blame it on your genes, or you have these kind of genes, right? And those are dependent on proteins. So proteins do a ton of things. So DNA is the molecule of inheritance, though. RNA has more to do with protein language. Okay, so here is a much better picture than what I drew previously. Here's our sugar. So the technical term for DNA is deoxyribonucleic acid. So this sugar is a deoxyribose. We have our phosphate over here. And then we have our nitrogenous base. And this one happens to be adenine, which is simplified as A. Okay, so here is our sugar phosphate backbone. Okay, and then we have our little nucleotides poking out because we're going to have another sugar phosphate backbone over here. And then more nucleotides that are going to join with each other and form that double bonded helix of DNA. Okay, so you see here we have our sugar phosphate backbone and then our little nucleotides, our little nucleic acid thingies joined in. Here we go. Our little nucleic acids joined here. Where'd it go? Okay, there we go. Joined together right here. You see one side. So we have our G, which is guanine. bonded to our cytosine, which is this C, and they have hydrogen bonds right here in the middle, keeping those different nucleotides together. Okay, so your genes are coming from DNA. So we take a little splice out of there, convert it to different types of things. So we have our gene, we take the DNA, we cut a piece out, it becomes RNA, that's called transcription. And then going from RNA to protein is something called translation, because you have to translate the language of nucleic acids over to a protein language, right? Because they don't really speak the same language. So this is translation. All right. And then you get your protein. And the proteins are tagged to be different things, of course, within our bodies. All right, so that's the end of this chapter 3. Go ahead and take the quiz after this. Make sure you take good notes just to make sure you're getting a good score on that quiz. All right, thanks for listening.